Thanks everyone for sticking around to the bitter end of the actual conference part. I'm super happy to be here. I've had some really I've learned quite a lot from all of you, so thank you for that. And thanks to Jennifer and the other organizers for inviting me. So I'm really excited today to have a chance to talk about some of the work in my lab that I actually don't often get to talk about. So this is hopefully going to be pretty fun. So my lab very broadly is interested in understanding the molecular mechanisms that support memory formation. So we ask kind of big picture questions about how certain types of memory work, and then we try to drill down into what mechanisms are supporting that memory at the gene expression level and at the level of epigenetic regulation of gene expression. And so one of the big picture questions I've been really interested in for a long time is how do existing memories update with new information, and how is that different from sort of de novo memory formation? Right, so you take a mouse, usually in the lab we take a mouse that has a very impoverished life, we give him this very salient experience that's really different from anything he's experienced before, and then we ask what mechanisms support that. But I think as humans, most of our memories are really modifications. They're updates to existing information we already know, and yet we understand comparatively less about how that process works. So we've known for a long time that when an animal initially learns something, there's a temporary window of consolidation during which that memory is stabilized before it's placed into long-term storage. So during this consolidation window, that memory is susceptible to a bunch of amnesic agents, right? It requires transcription, translation, a bunch of kinases, post-translational modifications. And if you manipulate any of those during this consolidation phase, you disrupt the memory. But once the memory reaches the storage phase, it's pretty resistant to disruption. That same amnesic agent has almost no effect. But if you give the animal a reminder cue, a retrieval event, that initiates a secondary process termed reconsolidation, during which, again, that memory is made labile. It's susceptible to a lot of the same amnesic agents before it's placed back into storage. Now, it was originally thought that sort of any reminder of training could initiate this process of reconsolidation. But in the past 10 or so years, it's become increasingly apparent that you actually have to present some new information to initiate reconsolidation. If you just give an identical training trial to a well-trained animal, that reconsolidation process is often not initiated. So one purpose of this reconsolidation process is thought to be to allow that memory to update. So I'm going to call this reconsolidation or reconsolidation-based updating to indicate that one purpose of reconsolidation is thought to allow this existing memory to update with new information. So, yeah.
So if you have a time gap between a well-trained animal and after a large time gap to the same exact time,
yeah, there are a lot of boundary conditions on reconsolidation for sure. Usually, if it's a remote memory, like 30 days later, sometimes that is not initiated unless you present something really new, but also linking that memory. But there are absolutely a bunch of boundary conditions on this, like time. So we have a bunch of outstanding questions about how this process works. So one question that I'll start off with is, can we leverage this process, this memory updating process, to simplify a memory, and what parts of the brain are important for that memory? We also want to know whether there are any distinct molecular mechanisms engaged by this reconsolidation updating process that are unique and are not dependent for the original memory itself. And then we also want to know what happens to the original memory in response to the update. What happens to that information? Is there competition? How does these pieces of information work together? So my story really begins when I was a graduate student with Fred Helmstetter at UW-Milwaukee, where I really became fascinated with this idea of memory updating. So in Fred's lab, we spent a lot of time studying fear conditioning as a model for memory. So you're all familiar with fear conditioning at this point for sure, but usually when people present fear conditioning, they're doing a delay version of fear conditioning. So you present an initially neutral conditional stimulus, in our case a white noise cue, something the animal is not afraid of, but then you pair that with an aversive stimulus, an unconditional stimulus like a shock, and it takes a single pairing for the animal to learn the white noise predicts the shock, and they start to freeze to the white noise cue. Now, in a delay model, the important part is that there's no gap in time between the offset of the white noise and the onset of the shock. They're presented contiguously, and it's very easy for an animal to learn this. So it's a very simple type of learning. It's largely subcortical. It depends on a very amygdala-centric circuit. And importantly, humans don't need contingency awareness to successfully learn delay fear conditioning. So in my graduate work, I was really interested in a variant of fear conditioning called trace fear conditioning. So in trace fear conditioning, you use the same two cues, the same white noise cue predicts the same shock, But we put a trace interval between these two cues. So we separate the white noise from the shock by about 20 seconds. And just adding in that little gap in time drastically changes what brain structures are necessary to learn this, and it makes the task much more difficult. So it's more complex and relational, as you can imagine. It's cortex dependent, unlike standard delay fear. And importantly, humans do require contingency awareness for successful learning. So we think trace fear conditioning is perhaps a better model of the complex and relational memories that underlie a bunch of different anxiety disorders. Yeah. Yeah, I mean, it's debatable for sure, but they've done a lot of studies where, for example, if you give like a distractor cue and you make it so that they can't explicitly say the white noise predicts the shock, for delay fear conditioning, they'll show an unconditional, like a skin conductance response to the delay cue, but they don't in trace unless they can explicitly say. Or if you modulate, if you do like a peri-threshold CS, so it's either just above or just below the threshold where the person can explicitly say they heard the CS. In delay fear conditioning, even on those trials where it's just below that threshold, they'll show that skin conductance response, but they won't for trace. So basically what I did in my dissertation work is I tried to figure out how these two types of memory extinguish and how they're different. So we know a lot about how delay fear memories extinguish, and to kind of oversimplify the circuit here, and sorry for this really ugly diagram, this was made before like BioRender was a thing. So you see that you need plasticity in the amygdala for successful extinction of a standard delay memory, as well as plasticity in the infralimbic medial prefrontal cortex. So for a trace memory, a lot of that plasticity is shifted up into the cortex. So instead of requiring this plasticity in the amygdala, you instead require plasticity in the retrosplenial cortex, which Megha had talked about this morning, as well as the prelimbic portion of the prefrontal cortex, and again, the infralimbic. So a lot of that subcortical plasticity is actually shifted up into the cortex for trace fear extinction, which means that this is a relatively complicated thing. We really don't understand the circuit that supports these complex relational memories. And what we do know is that it seems to be pretty complicated. This means that it might be relatively difficult to actually access these memories that rely on this kind of poorly defined, largely distributed cortical network. So this is kind of where this updating comes in. We had this kind of crazy idea that perhaps we could use memory updating to actually simplify the neural circuit that supports that complicated trace fear memory. What we thought we might try to do is train a bunch of animals with trace fear conditioning, give them an updating trial of delay fear conditioning, so just change when we give that shock. Instead of giving them a gap in time, now we present them contiguously for just one trial. And then we can ask, does that actually force this complicated memory to rely on that simpler, kind of more subcortical neural circuit? We figured if we could take this complicated kind of complex and relational memory and kind of force it to rely on the simpler neural circuit, we might make it more amenable to all of the treatments that have been developed with delay fear conditioning in mind, because most of the field is using delay fear conditioning to understand how this works. So what we decided to do is test this out using what I had learned in my dissertation about how these circuits are dissociable. So to just keep things very simple, we found this really nice dissociation between the role of the retrosplenial cortex and the role of the amygdala. So for the retrosplenial cortex, it's not important for delay fear conditioning or delay fear extinction. But for trace fear conditioning, you have to have the retrosplenial cortex for both extinction and even just the retrieval of the memory. And the amygdala, as I mentioned, is really critical for delay fear extinction, but we showed in a bunch of experiments that manipulating the amygdala had almost no effect on trace fear extinction. So what we can do is we can train animals with trace, update them with delay, and then probe these two regions to ask, are we actually changing what parts of the brain are important? Are we simplifying this memory? So in the first experiment, we asked if we take animals trained with trace conditioning, we update them to delay, is the amygdala now brought back on board? Do we have to have the amygdala for extinction? So the protocol is shown here. Basically, we train all the animals with that complex trace conditioning, and then we either update half the animals to delay, or we have half the animals that get no shift and they're just given one more trial of trace conditioning. And then we ask if the amygdala is required for extinction by just injecting AP5, an NMDA receptor antagonist, into the amygdala before extinction, and then we see if that extinction is affected. So I'm just going to show you data from the CS test. For the animals given ACSF, our vehicle control, we see very similar behavior whether these animals are not shifted or are shifted to that simpler delay. If we look at the animals that are not shifted, we replicated our previous finding that manipulating NMDA receptors in the amygdala has no effect on extinction of a trace memory. But if these animals are given just one trial of delay, now they show this effect where that extinction is impaired. So if you train animals with trace and you update them to delay, the amygdala is now again required for successful extinction. But perhaps, yeah.
So the X's in your upper right, that refers to the APD?
Yes, exactly, yep.
So it's not that you're lesioning it or inactivating it, that you're blocking plasticity or
Exactly. Exactly, yep. Yeah, so we decided to try the opposite as well, which is probably the more important. Yeah.
Blocking plasticity
During extinction, yep. Yes. This is a little bit different, but that last experiment was during extinction, yeah. So we asked, kind of the more important question perhaps is whether we're actually simplifying these memories, right? The problem is that they have this distributed cortical network that makes it really difficult to know where to look. So if we can actually shift where the memory is formed, perhaps we can make this distributed cortical network no longer required. So here we decided to test if after that updating process, is the retrosplenial cortex no longer required? So here, we train animals to trace. We either update them to delay or give them no shift, and then we ask if the retrosplenial cortex is necessary for retrieval. And in this case, yeah, we're injecting AP5 before that retrieval session. Yeah?
So just if you're doing a test, are you testing, it's hard to tell, right? You don't know whether they've formed a trace or a delay now.
Exactly, yes, and we'll get into that in a bit. Exactly, like the content of the memory is almost impossible to get at in this task, absolutely. Yeah, so we're kind of just using what we know about the circuitry to really try to figure out if we're actually moving the memory in the brain or moving what parts of the brain are necessary at this point. So we're just asking, do they need the retrosplenial cortex? So the trace memories require the retrosplenial cortex. Delay memories, retrosplenial cortex does not participate in. So here what we see is if they're not shifted, we replicate our finding that the retrosplenial cortex is critical. So you can't retrieve the memory if you have AP5 in the retrosplenial. But if these animals are given just one trial of delay, that manipulation has no effect. So just giving one trial of delay actually seems to change what parts of the brain are participating. And just to show you, this isn't a permanent effect. That memory does come back in our trace animals if we test them again after the drug is gone. Okay, so what this is suggesting is that if you give the animals a single updating trial of delay fear conditioning, it seems to actually change the parts of the brain that participate in retrieving or extinguishing this memory. So we can take, in theory, this relatively complicated circuit and make it simpler using these updating principles. But as you mentioned, we really can't tell with this task if the content of the memory is updated. So as my grad advisor would say, rats are pessimists. They kind of freeze for the duration of the trial. It's not carefully timed. You can't tell if a rat is freezing because it's afraid of a delay configuration or a trace configuration. So we really wanted to ask some of these questions about what's happening with the content of the memory. So when I started my postdoc, I actually developed a new task to look at kind of the content of the memory as well as some of these mechanisms. So the task that we developed, we call the OWL paradigm, or the objects and updated locations paradigm. And this is based on object location memory, which we've heard a little bit about. But the beauty of this task is that it's a very simple spatial task, so it's exquisitely dependent on the hippocampus. It's an incidental learning task, so these animals are kind of programmed to study anything that's novel. It's capitalizing on what these animals are really good at doing anyway. And there's no aversion or repetitive information presented. It's kind of a very neutral type of learning that's probably very similar to a lot of our sort of everyday memories. We can play around with how well they learn these things. So in this task, the animals are learning where two objects are located in space. So we get the animal very familiar with this context, and then on the training day, we present two identical objects in very specific locations in this environment. So we use 100 ml beakers filled with cement. They're identical objects. And so we want to know if the animals are learning where these objects are located in space. And so to test that in a normal object location memory task, you would have what you see down here, where you move one object to a new location. So mice are driven to explore anything that seems novel. So if the animal remembers where these objects were during training, this moved object should seem new and interesting, and we should see preferential exploration there. And of course, so really what we're trying to get at here is whether the animal is learning this information about where this object could be located. So do they actually update this memory with this new information that an object could also be located in A3 here? So in order to test that information, we now give a test session the following day where we expose the animal to 4 objects. 3 of these have previously been presented, A1 and A2 during the original task, A3 is the update, and then A4 is in a novel location the animal's never seen before. This allows us to sort of index whether the animal remembers each of those familiar locations by comparing to a novel location, right? So we have our original locations, our updated location, and then one in a novel location. So we can ask, is the original memory intact by comparing exploration of A4 to A1 and A2, and we can ask if they've learned the update by comparing A3 to A4. So let me show you what it looks like when an animal does this task, because we were worried. We had switched to mice when I was in my postdoc lab, and we were very concerned the animals wouldn't be able to do this. They're maybe not engaged by the task the whole time, but they show really nice memory in this task. So first, I'll walk you through what it looks like in each phase of the experiment, but what I'm going to show you is a discrimination index. I should have put my equation on here to show you that I do a little bit of math. But in the discrimination index, we're just calculating a preference score. Does the animal prefer one object over the other? So during this initial training session, these objects are equally novel. The animal spends equal times exploring both of those, whether they're designated in the no update group or the update group. And then we also look at total exploration. So in that session, how much time is the animal interacting with those objects? Okay, so we see no preference during the training session. If we now look during the update session, this is our no update group, and they again show no preference between those now familiar objects. But if we move one object to a new location, this is just standard object location memory at this point, you see the animals prefer the moved object compared to the unmoved object. And we do see a slight uptick in exploration for these animals that are given this new location. They're probably more motivated to explore. Yeah.
So is it exactly the same object?
Exactly, yep. Actually in this task, in this version, we actually replace the object because we can't possibly use the same object in 4 locations for the task, but they're identical. The animals can't seem to tell them apart. Okay, so then if we look during the test session, we get information from the original object location. So here I'm showing you location A1 compared to location A4. This is the original object presented up here that is presented on all 3 days of this session. And then here I'm showing you location A2, which is presented on the training day, and then it's either presented for the no update group or it's absent for the update group. So what you can see is the animals are all showing a preference for location A4 compared to location A1 or A2. And you'll notice that for location A2, we do see a slight decrease in memory for our updated animals, which we pretty reliably see. And we think that's because there's actually a 48-hour retention interval for these animals, because the update animals don't see location A2 on this second day. They don't see it since the training day. So it's actually a little bit more sensitive sometimes to some of the manipulations we're doing. Yeah. The no update group just gets one more day of training, the same 2 objects, A1 and A2, and then the update gets A1 and A3. So one object is moved to a new location. That make sense? The no update's just kind of our control. They get like essentially one more day of training with nothing changed. Yeah.
And how do you get A4? How do you get A2?
Yeah, so we just kind of name these objects. So if you look at the test session, we call A1 and A2 are the 2 objects locations presented during the original training. Location A3 is the updated location down here. And then A4, they've never seen an object in that location before, and that's our novel location to compare everything against. Yeah, okay.
So how is this connected with delay and trace?
It's not. It's we were really trying to probe the content of the memory now. So it's a very different. They're trying to learn the locations of those objects, essentially. No, they're just, it's kind of taking advantage of their natural preference to learn where objects are located in space. It's an incidental learning task, but over a 10-minute training session, they'll learn where those objects are located. It's just kind of a standard spatial memory. So then we look at the updated location, and we see the no update animals show pretty equal exploration of those 2 objects. Again, so at this point, the no update group has never seen A3 or A4, so they're equally novel here. But you'll notice that if we compare A4 and A3, now the animals that were exposed to location A3 in the past, they show memory for that, and they prefer that novel location. And then we can just look at total exploration. So we have this nice task where in the test session, we can really look at the original locations, the updated locations, and figure out if they've updated the memory, what that does to the original information, etc. Okay. So we spent a lot of time kind of validating this task. Yeah?
I'm just a little confused by the term
Yeah.
Is it just three separate memories now?
Great question. Yeah, we spent a lot of time actually trying to validate that this wasn't a brand new memory, it was actually an update of the existing information. And of course, it's a little bit arbitrary, right? Like most memories are linked in some way to other things, but we have two separate lines of evidence that instead of being a separate memory between the trained information and the update, that these are really, it's actually an update of the existing information. So one of the things that we've done is we came in with anisomycin infusions. So we know protein synthesis is required for both, for that reconsolidation process. And one thing that you see is in a reconsolidation paradigm that the memory is labelized because it's brought out of storage and thought to update. So one of the things we did is we gave an anisomycin infusion after that update session, and we showed that that doesn't just block the updated information, it actually impairs the original memory itself, suggesting that the original memory is made labile in response to this updating task. And the other thing we did is we used ARC-based catfish to look at the neuronal ensembles that are engaged by the original information and the updated session, and we show that the original ensemble is preferentially reactivated by that update session. So we're pretty convinced that it's really a memory update as opposed to a brand new memory that's just maybe competing for expression like an extinction memory. And if you are interested in this task, I mean, I think this is a really very simple paradigm to set up. We do have a procedure paper that a really wonderful, talented undergrad in the lab who's now doing a postbac has published to really walk through all of the protocol here. Do you have a question? Yeah.
You say that the age is also updated. So that is basically the second training is taken away. That also maybe explains why actually in the update group,
Yes.
like the A4 was actually less preferred, right?
Yep. Yeah, absolutely. Right. Yeah. We typically say like, oh, there's a longer retention interval, so they're maybe starting to forget it faster, but you're absolutely right that they're also exposed to not having an object there, so they're probably learning this object is not always present, as opposed to like A1, which is present and never moves throughout every single session. Absolutely. Yeah, Jennifer.
Have you played around with more exposure to the original configuration because it was 10 minutes, 5 minutes?
Mm-hmm.
What if you had like, you know, tenfold more exposure to the first and then you do one?
Yeah, we have done, so we do this in old animals, as I'll show in a minute, and to do that we have to kind of boost the amount of training. So we've done that directly comparing young and old animals with 3 training sessions, and it looks remarkably identical. But that said, I think that you can overtrain the original memory and make it so it like resists updating and things like that. Absolutely. Yeah. Okay, so we're really curious about using this paradigm to answer questions about how updating works. And one thing we're really curious about is how this process is impaired in old age. So we know that aging is accompanied by memory inflexibility as well as susceptibility to proactive interference. We know that aging animals struggle to learn original information, but we also know that once that memory is formed, it's pretty resistant to being updated. So we wanted to see if we could pick up on this using our task. So here we're comparing young mice as well as old mice that have normal impairments in memory formation. And so, as I mentioned to Jennifer's question, we modified the paradigm to make sure the old animals could learn this task. So we give them 3 successive days of training of locations A1 and A2. We want to really make sure they learn the original memory before we try to modify it. And then we either give them no update or an update and then test them as we've done before. And so what we see is that first, if you look during the update session, this is going to give you an idea of whether they learned the original locations. If they prefer the moved object, that indicates they have memory for that original training session. And so here you can see when the animals are updated, both the young animals and the old animals, they show robust memory for the task. So the white bars are animals in the no update condition, the gray bars are animals that were exposed to location A3, and if they were exposed to A3, they prefer that object because they recognize it's novel. But we see, again, a slight increase in the young animals exposed to that updated information. The old animals, we see maybe a trend in that direction, but they largely explore a little bit less, because they're aging. Okay, so then if we look during the test session, if we look at the original location A1, they all show robust, intact memory for that location. For A2, we see the young animals with that 3-day training protocol show robust memory even for location A2, whereas the old animals are starting to show a decrement in that memory. But when we look at location A3 and we ask, were they able to update that memory? We see the young animals show robust memory updating, but the old animals are not preferring location A3. It's as though they had never been exposed to that location before, and they're showing no evidence of memory for that location. And again, we see slightly less exploration in our old animals, but we don't see any difference based on whether or not they're updated. So we see that this memory updating process seems to be impaired in old age. And as I mentioned, my lab is really interested in the molecular mechanisms that support this. So we're actually running right now some RNA-seq studies to try to figure out in an unbiased manner what are all of the genes that are messed up with aging or that are affected by this updating process uniquely. But one of the things we decided to tackle kind of in the immediate term is looking at a repressive histone deacetylase called HDAC3 that we previously have shown is critical for memory formation but impaired in old age. So HDAC3 is a repressive histone deacetylase. Its job is to remove acetyl groups from a histone tail to keep a closed chromatin structure, making it really difficult for gene expression to occur. And a lot of previous work I did as a postdoc showed that if you get rid of HDAC3 in the hippocampus, you create a really robust memory in young animals, one that's usually more persistent than a regular memory. And in old animals, if you get rid of HDAC3, you can ameliorate age-related memory deficits. So we said, we know HDAC3 is really critical for memory formation. Is it also necessary for this updating process? So Chad Smeeze, who's really taken over this project along with Destiny, they ran an experiment where they took old mice, they trained them over 3 days to those 2 locations, they give them that update session, and then they inject an HDAC3 inhibitor. So RGFP966 is a selective HDAC3 inhibitor. So they do it after the update session, and then they ask if that memory is intact the next day. So before they get the drug injection, during that update session, we see these old animals, again, are able to learn the original information as long as we give them 3 training sessions. When we look during the test session, we see intact memory for the original information. And then when we look at the update, we see the vehicle animals struggle to learn that updated information, but blocking HDAC3 ameliorates this deficit, and now these animals show robust memory for that update without any effect on total exploration. So it seems like HDAC3 is contributing to this age-related memory impairment. So we thought we would run a kind of a quick control experiment just to see what this is doing in the young animal as well. So we ran a very similar experiment in young animals. Here they're only giving 1 day of training, however, and that'll be important in a minute. So they give 1 day of training, they either no update or update, and then give that RGFP966 injection and ask what happens to the memory. And so what they found is we see normal updating. This group does have a no, I should mention, that last experiment we did not have a no update group just because these old animals are kind of difficult to acquire. We try to run it whenever we can, and so here we have a no update condition versus an update. And if we look at the test session, if we first look at the updated information, we see that blocking HDAC3 in the young animal that's already learning this update has really no effect. And we weren't super surprised by this. The animals are at a ceiling at about 25 to 30 for a DI here. So we didn't see any further enhancement of the memory, which wasn't surprising. What was surprising is when we looked at the original memory itself, it's intact in the no update group, but in the update condition, we see that it's actually slightly impaired when we block HDAC3 after the update. So it's like blocking HDAC3 after the update session is perhaps strengthening the updated information at the expense of the original information. So we decided to figure out, we're trying now to figure out why that is. Why is RGFP having a different effect in young and old animals? So our hypothesis is that the original memory and the updated information are effectively competing for expression. And when you give HDAC3 inhibition, you're shifting this balance. So when you take an old animal that doesn't learn the update and you block HDAC3, that's strengthening the update information so it can effectively compete for expression with the original information, but they're both expressed at test. In the young animal, these animals are already learning the update just fine. So when you combine that well-learned update with an injection of this HDAC3 inhibitor, you're effectively allowing this update memory to dominate or maybe saturate the memory at the expense of the original information. So we decided to test this by manipulating the strength of the original memory and the update to see if we can play with that balance. So what we did originally is we injected that RGFP966 after the update session, effectively enhancing the update or strengthening the update at the expense of the original information. But if we give, say, a sub-threshold update, we give them just maybe 1 minute of exposure with that updated information, perhaps we can restore that balance so both memories are expressed at test. Alternatively, if we strengthen the training session by giving them 3 training trials here, perhaps we can strengthen that memory enough so that it's able to persist and be expressed at the test session. And then finally, if we change when we give that HDAC3 inhibitor, maybe we can do the opposite and create a really strong saturated training memory that effectively prevents updating. So we're still working our way through these, but I do have data from 2 of those 3 experiments. So first, what happens if we give a really weak update? So we're kind of mimicking what happens in the old animals. They don't normally update because it's not sufficient to drive that updating process. So what if we mimic that in our young animals, give them just a single minute, a subthreshold update session, but then strengthen it pharmacologically by blocking HDAC3? So I'm not going to show data from the update session for this experiment. It's quite hard to interpret what's happening in just the first minute of an update session. But what happens during the test session is interesting. So we replicate that, we had kind of run a series of experiments to verify that 1 minute was not sufficient for updating, and we replicate that here. And you see the vehicle animals are not reliably showing memory for this update. But if we give that HDAC3 inhibitor in the young animal, now they show memory updating. And when we look back at the original memory, if we weaken the update, now we see that that memory is still intact and is not weakened in response to HDAC3 inhibition. And again, we see no difference in total exploration. So playing with the strength of the original memory in the update seems like if you give a weaker update and combine that with HDAC3 inhibition, you can restore that balance. And then finally, we tried the opposite. So we asked what happens if we give this HDAC3 inhibitor after the training session instead? Is that sufficient to sort of make a really strong memory for the original information that can no longer update? Okay, so when we do this, we see intact memory for the original information. And then if we look during the test session, you see the original information is intact as you would expect. But again, if you give HDAC3 inhibition after that original training session, those animals that got the HDAC3 inhibitor around the time of training are showing deficits in the ability to update. So they're not able to learn that updated location. So what this suggests is that reconsolidation-based memory updating may be sufficient to change both the neural circuitry supporting a memory as well as the content of the memory, and that HDAC3 may be contributing to age-related impairments in memory updating. And there seems to be this competition between the original information and the update. And if you strengthen one, if you kind of overtrain or overstrengthen one of those, the original information or the update, they seem to compete for expression. You may impair the ability to update or the ability to recall the original information. So with that, I'd like to thank my lab. I talked a lot about Chad Sneeze and Destiny here, as well as all of you for your attention. So thank you.
Maybe I missed this, but what is the evidence that it's updating of the memory versus competition of two distinct memories? And you had some neural representation evidence, but what is the strongest case that it's not just two separate memories competing for control of behavior?
Yeah, I actually think, when we inhibit HDAC3, we might be actually creating, we don't know if the mechanisms are the same as under the original paradigm, but when we look at just the standard paradigm, our evidence that this is actually a memory update as opposed to two separate memories. The first thing we did is we blocked anisomycin after the update, and we showed that that not only prevents the actual update from being learned, it impairs the original memory itself, suggesting that that original memory is brought out of storage and made labile in response to the update. But we also looked at ERK-based catfish to look at the neuronal ensembles involved in the original memory versus the update, and we showed that there's much more overlap when we do that versus when we do a new memory. So we're pretty convinced under natural circumstances this is an actual update, but I don't know if when we block HDAC3, perhaps we're biasing maybe a new group of neurons to come on board. We haven't looked at that yet.
I'm wondering too if you could get at that by changing the object as well as the place to really use some combination. I can't quite design it online here, but to make behavioral evidence for it being an update rather than...
Yeah, I mean, we have, so when we did that ARC stuff, we actually compared, an update where we just move one object to putting them in a new context and giving them completely different objects, basically. We showed that that has much less overlap, absolutely.
Thanks. I think the idea about the differential contributions of retrosplenial versus amygdala plasticity are really interesting and important. Yeah. Your discrimination index for your behavioral analysis, that's essentially defined as the differential interaction times, right? Yeah. What's your definition of interaction?
Yeah, so we have a very precise definition of what counts as like object exploration. They have to be within a centimeter of the objects, and nose has to be oriented toward the center. And we don't count it if the animal's rearing because we can't tell if they're actually engaging with the object or if they're like, sampling the environment effectively. And we have a pretty like stringent set of criteria for that. Because we see like a ton of variability when the criteria aren't stringent like that.
Sure. I mean, I'm sure it's empirically based, but
Yeah.
You know, probably some of this at least could be visual and or olfactory.
Yeah.
And maybe the older animals by the end, by U4, could be moving to a different sort of, you know, interaction mode.
Sure. Something that I didn't mention that I think is interesting and gets at this question is we did look at short-term memory for both young and old animals. And the old animals do show memory for the update if you test it an hour later. They just don't reconsolidate that into long-term storage. So they seem to be motivated to do the task, seem to have the skills to learn where those objects are located. They're just not restoring that information.
Thank you. Actually, quite related to Robert's point. Very clear talk. Thank you. This is really not my field, so I'm asking a maybe dumb procedural question. I was looking at the timescales of the exploration, and I may have misread, but it looks like it's lower than 5 seconds in total. So if the training session is minutes long, and the total exploration times is 5 seconds, the first question is, how robust are you finding across mice? And then what are the mice doing the rest of the time? And is that something that, you know, as Mate was saying, we're using a scalar measure of performance, but maybe they're just sitting in the middle and just looking around and they're learning that way. So I just don't quite understand really the robustness of it.
We have, like I said, we have a pretty stringent criteria for what actually considers exploration. So we do typically see maybe 10 to 15 seconds of exploration out of like a 5-minute session, which is relatively low. They're near the objects a lot. We do 6 full days of habituation before we even start this, because mice are afraid of everything. And if you don't do that, they just won't interact with the objects at all. So we do a lot of explorations that they're able to like explore the environment. They're comfortable there. They spend a lot of time rearing, digging in the bedding, doing other mousy things. But, you know, that is reliably enough time for these animals to learn the task. It's like they're actively exploring it during that time, and that's sufficient for them to form a long-term memory for sure. I had something along that line that I forgot. But yeah, I mean, it's definitely sufficient for them to learn the task. And they do spend a lot of time doing other things, but they definitely they have a very distinct, I should have shown a video, very distinct behavior when they actually explore with the object. It's almost like there's a magnet on their nose drawn to the center of the object. And you can really, when you get good at looking at mouse behavior, identify it pretty simply.
Related to Elisa's question, when you have 4 objects, you're not only changing the position of the objects, you also have more objects in the room, so there will be contextual changes. Do you have any way of measuring what the animals are doing in that other time, the rest of the time, and do you see differences between the old and the young mice?
You know, we have all the videos, and in theory we could put them through Deep Lab Cut or something and see if we can identify, like, you know, I assume the old animals do less rearing, they definitely move less, like they're obviously less limber and, and excited to be moving around, but it's possible that maybe the animals who don't update show some other behavior that, you know, we aren't picking up with just like our basic scoring protocol now. So it might be worth analyzing those data with some machine learning or something to see if we can identify any other behavioral pattern that indicates whether or not, like, that maybe matches up with what we're seeing with this measure. That would be really interesting, but we haven't done that.
So also related to Mate's talk, do you have a measure of saving? So, for instance, if you then you re-update the memory back to A2, are they then forced to learn it?
Yeah, we haven't looked at salient. We haven't tried replacing A2 to see if, I mean, they clearly remember there was an object there. Like they show more memory for A2 than A4 usually. So they clearly like still remember that information. We haven't looked at, salience besides the fact that A2 is a little bit older. I'm trying to think of how you would look at salience here besides maybe changing, as Jennifer suggested, the identity of the object and maybe making something that they really like exploring or making it appetitive in some way. But of course that changes the motivation and it might make it difficult to interpret.
But compared to your fear conditioning, so there you were sort of arguing that it's really gone away, right? The substrate has really completely changed. And that's not the case in your updating, in your ALD protocol.
Right. Yeah. That's absolutely true. And we certainly haven't looked at like obviously all this update stuff is all happening in the hippocampus too. I would love to do some of those questions about whether the memories actually changing like at a neural circuit level, but we really know very little about the neural circuit supporting object location memory, except that the hippocampus is really critical, and we've looked at retrosplenial as well.
in a similar vein, and this is very interesting because we've started doing some of this type of task and figuring out how to analyze the data. But it just occurred to me based on your presentation and the question before, you could do sort of a quadrant analysis, how much time they spend in the quadrants of the box where the animals are. And it'd be interesting to know whether they actually, once you've shifted the the object in A2 to A3, whether they actually go back and explore in A2, for example, more than maybe around A1, as another kind of sign of memory, I suppose, in terms of something being different. Not only is there an object in a new place, but now there's no object in a previous place.
Actually, I have a little anecdotal. We don't usually look at a quadrant analysis just because it's not quite precise enough for us to see like bone. Like they explore the objects a little bit differently than if we compare like how much time they spend in quadrant. But anecdotally, I had an undergrad who accidentally gave habituation with 2 objects in there because she didn't know what she was doing. Obviously a supervisor issue though. But yeah, so we decided to train them just to see what would happen with putting the object in like the bottom corner instead. And they did spend much more time exploring where those objects used to be. So I would expect if you did a quadrant analysis or even drew an invisible circle around where that object used to be, I bet you they do spend more time kind of trying to figure out why it's not there. So to your point as well, like they probably are updating, like this information is, it's a prediction error, right? There was something there, now there's not. That's also something to learn. Yeah, that's a good point.
And just a couple of procedural things. How old were the old mice? And are you using male and female mice?
Yes, they're 18 to 20 months old. We are using male and female mice. We don't typically see any differences. And we've seen very similar behavior. So 18 to 20-month-old mice are pretty reliably impaired in the initial formation as well, which is why we chose that age.
All right, maybe I'll jump in with a question. So I'm just curious, I can't quite make the relationship in my mind, and maybe there's not one, but is this idea of like competition domination, do you think that's related to retrieval-induced forgetting and sort of the non-monotonic plasticity hypothesis?
Yeah, it's definitely possible. We really don't know at this point. I like to think that perhaps like HDAC3 is sort of releasing this molecular brake pad. So whatever the neuron is doing around that time, it's getting very engaged with. It's getting this massive wave, presumably of transcription that'll ultimately be translation. All these synaptic changes are really blasted on the cell. So I think if that's happening during training, I think that it's almost like saturating those cells so they are less modifiable for the subsequent update and vice versa. They have maybe a weak memory, but they're activated by the update session. With that HDAC3 kind of releasing the brake pad, they're just sort of committed to this new information perhaps. But we're still trying to figure out exactly what's happening. We'd love to partner with any electrophysiologist to maybe look at some plasticity things. But yeah, I mean, we're mostly focused at a molecular level to try to figure out what exactly is happening there. It's a great question.
Any last questions? All right, let's thank Janine.
Thank you.